Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Yvette Wong, Assistant Professor of Neurology at Northwestern University's Feinberg School of Medicine. Yvette is also a 2024 Toffler Scholar. Today, we will explore Yvette's groundbreaking work on organelle dynamics, neurodegeneration, and her journey as a scientist. So welcome, Yvette.
Hi, it's really great to be on this.
So let's just start with kind of explaining what is an organelle?
Yeah, so organelles are basically the components of things inside cells or neurons. And so examples of organelles are mitochondria, the powerhouse of the cell, or lysosomes, which help to degrade things inside the cell. The nucleus, which contains all our DNA, is also an example of an organelle. So every cell contains lots of different types of organelles. And for things such as mitochondria, they can contain hundreds or thousands of them.
So those are different. Organelles are different than organoids, because I get those two kind of mixed up, because that's another term we're starting to hear. Can you describe the difference?
That's right. So organelles are components inside the cell, whereas organoids are more commonly now used as a term of a collection of cells that you can grow, for example, in a dish to sort of mimic an actual organ in the human body. So an organoid would be a large collection of cells altogether, and the organelles are the subcomponents of each of those cells.
So how did you get started in looking inside like this microscopic look inside a cell? What was that journey?
Yeah, so it started when I was a PhD student. And at that point, I started a project where we were looking at an organelle known as an autophagosome. And we could basically look at the dynamics and the trafficking of these autophagosomes inside axons and watch their transport over time. And so that really made me fascinated about the fact that we can track these organelles and their dynamics over time in live cells or live neurons. And that was really exciting.
So why is that important to kind of track this movement? What is it telling us about the brain or diseases? Why is that important?
So we think this is important for several reasons. First, we know that in neurons, they have these extremely long axons. And so the axonal transport of these organelles down the axon are really important for neuronal function. There are a lot of mutations in different proteins that regulate these transport, and those mutations can cause neurological disorders. But in addition to this axonal transport of organelles, there's also this really interesting crosstalk between different types of organelles where they can actually contact each other at these contact sites. And so that's an interesting dynamic that we're really starting to understand more of as well.
And specifically, you're looking at, I think, two of these kind of interactions, lysosomes and the mitochondria. Is that correct?
That's right. Yeah. So we've been really interested in the crosstalk between mitochondria and lysosomes. We found that they are able to form these contact sites with one another. This was work when I was a postdoctoral fellow. And what was really interesting was at the time, the majority of crosstalk was sort of thought to be the lysosome helping to degrade damaged mitochondria in processes such as mitophagy. But the idea that perhaps there could be an important crosstalk just at an interface where the two organelles formed a contact site was still being explored. And so it was really exciting to see that, mitochondria and lysosomes can form these extremely dynamic contact sites. And these contact sites we're starting to learn have important roles in regulating both mitochondrial function and dynamics, as well as lysosomal function and dynamics.
I think you've described it as like a cellular conversation. And I think that that's a nice visual. Can you just kind of expand on that a little bit more? Like, what are they talking about?
Yeah, so we think there's actually lots of different things they could be talking about when they come together. So at different levels, we think one thing that could happen is that the mitochondria could be transferring cargo to the lysosomes and vice versa. And we and others have shown, for example, calcium can be transferred from the lysosomes to the mitochondria at a mitochondrial lysosome contact site. The second thing we think that is important is that it basically allows the proteins on the membranes of each of these organelles to directly meet one another. And so that allows, for example, a mitochondrial protein to interact with a lysosomal membrane protein and either drive an enzymatic reaction or regulate its binding with another protein. And so not only transfer of cargo, but also the regulation of actual proteins on the membranes of each of these we think is important.
And I think you, some of the recent, I think you had maybe a breakthrough or an understanding that when these contacts or this communication is disrupted, it may lead to, disease like ALS or possibly Parkinson's. Am I understanding that correctly?
Yeah, so starting from my postdoctoral work, we've shown that defects in these mitochondrial lysosome contact sites might be disrupted to various neurological diseases. So, for example, we found that in Parkinson's disease, mutations in Parkin or GBA might disrupt these contact sites and their function. And the other disease we've looked at is Charcot-Marie-Tooth disease. That's a genetic peripheral neuropathy. And in that case, we think actually one of the mutations that is disrupted in that disease is actually one of the main regulators of mitochondrial lysosome contact site tethering, which is RAB7.
So what's the next step then with this information? Is there, I mean, can you jump to trying to understand therapeutics, or can you look at kind of repurposing drugs? With this information, what's the kind of the next steps?
Yeah, so there's a lot of exciting next steps to this. One angle, as I mentioned, if we're looking at the transfer of cargo between these contact sites, if we discover that in certain diseases that this transfer is disrupted, we could then figure out how to maybe increase or accelerate this transfer to compensate. Or we could find, potentially there are other organelles that can deliver that cargo instead. We could maybe target the transfer from a different organelle to rescue that defect. And if we're thinking about the proteins on the membranes that are talking to one another at these contact sites, in those cases, the more we learn about those proteins, what complexes they form, and what are the enzymatic activities or functions that happen at these contact sites, we could potentially, think of different therapeutics to directly target those, whether it's their binding or their activity.
That's great. I think in a previous discussion I had with you, you gave kind of a visual of people on a bus and they're passing information, but there's people outside the bus and they're passing information to the bus. Can you expand that visual to really kind of go a little deeper in that discussion? Because it's hard for someone like me to understand what is this cargo and why are they communicating, because they're very active. These cells are very active. But can you just kind of expand that a little bit more?
Yeah. So one way I like to think about the cell is that it's a town and every protein in the cell, and there's, so many types of proteins, and each of them is basically a person and they all have an important function, but they can only be at one place at one time. And so where they are and when that happens is very important. And now with live microscopy, we can get a better idea of, where each protein is at different times. And so one interesting thing is that, we think of organelles as these highly dynamic things that can have lots of proteins on their membrane as well as inside them. And so one way to think of these constantly moving organelles are a bus full of lots of people. And so you might have one bus, which is the mitochondria, and another bus, which is the lysosome. And keeping in mind that, there are hundreds and thousands of each of these mitochondria and lysosomes in different cells. And so one way you could maybe have people on the mitochondria bus talk to the people on the lysosome bus would be if those two buses, drove up to next to one another and could then, sort of temporarily park beside one another at what we're calling a contact site, and then allow for the people on the bus, these proteins to, either talk to one another or potentially transfer cargo, such as, calcium or amino acids or different lipids from one bus to another to help each other's bus and their function.
Yeah, that's amazing. That's so helpful because it gives people like me just a visual of what is going on at a very, very small level. In talking about the small level, you use some really advanced technology to do your research and your work. You've mentioned some of them, but I think this super resolution live cell microscope. Like, talk about what that is and what other technologies are you using to see these buses of little mitochondria and lysosomes.
Yeah, this has been really fun for us. Like I said, when I was a PhD student, was when I first got really excited and introduced to the fact that there is such a thing as live cell microscopy. You can actually visualize, proteins and mitochondria and lysosomes moving inside a neuron. And so when I started my lab, I knew like the thing we really want to do is do this live cell microscopy. And, now there's really advanced techniques to do this. And one of the ways to do this is using super resolution microscopy. And so that basically means that we can look even more in detail inside a cell or a neuron, and we can track different proteins, and we can track different organelles such as mitochondria. And this has been really fun for several reasons. In the past, it was pretty hard to, clearly see what's going on inside a mitochondria. You could sort of, see the bus move, but you couldn't really tell what's going on inside the bus. And so now with this sort of microscopy, we can actually track some of these dynamics over time. And that's been really fun to see, how do these things change if we disrupt mitochondrial function? How do these things change in different disease mutations? And it's also been interesting because now we can track lots of different disease-related proteins and see how do their dynamics get altered over time.
So these advanced microscopes, it must create a lot of data too, because if you're watching things move, it must be a lot of data. And so maybe explain a little bit more. Are you using AI, or how do you get through the data to really get your understanding?
Yeah, that's a great question. So we definitely generate tons of data every week. We're constantly taking really cool movies. And a lot of the times it's movies that of proteins no one has ever looked at over time. So that's really exciting. And yes, there's a lot to process. So there's sort of two ways we're trying to get around it. One is, coming at it sort of with a hypothesis, like what change in dynamics are we hoping to see based on what we know about, other similar proteins or what we think might go wrong in this disease. That sort of helps us focus in a little on like what the dynamic change we should even be looking at or quantifying. And in those cases, we haven't used AI directly, but we have started using some sort of more advanced analysis to help us analyze these changes in the different cells or neurons we're looking at. And then the second approach that we're doing this is to sort of like target the proteins a little bit more systematically. Like I said, there's so many different types of proteins inside a cell. We could spend hundreds of years sort of looking at each one and how each of those change over time in different conditions and with different mutations. And so when we started the lab, we've basically looked at the really important proteins that are disrupted in these major neurodegenerative diseases to see, if we look at changes in their dynamics, does that give us a clue into the disease process?
Yeah, I think that as you were talking, it's that change detection just seems like that's going to be really important to really understand how the disease happens. So that's really cool. So I think through this process, you've identified some really interesting information around the Huntington's disease. can you kind of just start with what is Huntington's and what is your research with that hypothesis that you were maybe studying? And again, what are the implications for the next steps?
Yeah, so this is actually work from when I was a PhD student looking at Huntington's disease. And so Huntington's disease is a genetic neurological disorder. It's caused by a polyglutamine expansion in the protein Huntington. And because it's genetic, basically it's mutations of this polyQ. Anybody who has repeats of around 30 to 50 or even more than that will get Huntington's disease. And so what we previously found when I was a graduate student was that these mutations in Huntington's disease disrupt autophagosome axonal transport. And so that was really interesting at the time because there were only a few regulators of autophagosome axonal transport known. And so this suggests that perhaps these expansions in the Huntington protein could be really important in disrupting autophagy, which is mediated by this organelle known as the autophagosome. And that's important because one of the main roles of autophagy is to clear lots of protein aggregates and things like damaged mitochondria. And so potentially one of the many things that might go wrong in Huntington's disease is that autophagosome dynamics might be disrupted, leading to this buildup of damaged cargo or aggregates.
So and so you're continuing to study this, is that correct?
Yeah, so we're starting to look at some of the other types of polyglutamine expansions, as well as other neurological disorders that are also related to mutations with expansions. And one of those is a C9orf72, where mutations in that link to ALS.
So how close are we to, we're very far from a therapeutic, I would imagine, but how far are we from really understanding to where we could even start to consider what therapeutics might be delivered or developed?
Yeah, so I think this looks different for every disease. I would say in Huntington's disease, in some ways, this is actually a little easier because it's a monogenic disorder. So it's everyone with Huntington's disease has mutations in the exact same protein, the gene Huntington, and we know exactly what that type of mutation is. And so I think one of the ways people are trying to target this for Huntington's disease is to directly target the gene and to replace potentially any mutant alleles with a wild type allele so that they would have a wild type version of Huntington rather than this mutant version. So I think that looks promising so far, but I think for a lot of these other neurological disorders where it's not always a clear genetic cause, I think there's still a lot of work that we're still doing.
Yeah, which leads me to some of I think you're also studying ALS, and why did you kind of pivot there? What was that kind of pathway that you said, oh, there's some commonality here, or oh, I'm interested, and what are you learning?
Yeah, so ALS is a really interesting disease from a cellular point of view because there are a lot of different mutations that are known to cause ALS. And what's interesting is that of all these different mutations that are found in different genes, these proteins that have these mutations localize to all sorts of different organelles. And so, compared to Huntington's, where it's just one protein, one gene, in ALS, there's a whole bunch of these. And there's also patients who have no mutations, known as sporadic ALS. And in those cases, we're still figuring out, how do those patients end up with ALS? And so actually, when I was a graduate student, I had started working on ALS on a gene known as optineurin. And so mutations in optineurin can cause ALS. And at the time, we showed that actually one of its functions that is disrupted in ALS is its ability to regulate mitochondrial turnover. And so when I started my lab, that was one of the things I've still thought about. Basically, how can you have all these different proteins that are located at different organelles? And the interesting thing is that, now we know that there are these interorganelle contact sites. And so one possibility is maybe these different contact sites contribute to what goes on in ALS. And so there are, like I said, many classes of genes in ALS, but two sort of classes that are thought to be important are genes that are regulating RNA granules and also genes that regulating mitochondrial function. And so something that we're now really excited about in the lab based on some of our recent data suggests that maybe there's this really interesting crosstalk directly between RNA granules and mitochondria. And we think we've identified potentially some interesting proteins regulating that.
So, what is an RNA granular? Is there a relationship to DNA? What can you help me with that?
Yeah, so the nucleus is sort of the home for all our chromosomes, which is our DNA. And in the nucleus, the DNA will get turned into RNA. And that RNA has to leave the nucleus and end up in the rest of the cell. And that's where it can get turned into protein. And obviously, those proteins are used for lots of different functions to make up everything we have in our cell. However, one of the important organelles is mitochondria, and mitochondria also need to get some of these proteins that come from nuclear DNA. And so we think it's really interesting that potentially one of the ways that this happens is through these RNA granules. So the RNA basically leaves the nucleus, and once it's in the cytoplasm, it can interact with different proteins and it can form what's sort of called like a membraneless organelle. So it's not a classical organelle, which usually have membranes like mitochondria or lysosomes, but these RNA granules are still considered an organelle because they sort of stick together and they're a composition of RNA with proteins. And you can track and visualize these over time. And they move in the same way that other types of organelles can also dynamically be moving.
Yeah, as you're going through this, it is so amazing to me that people go into the doctor, they get diagnosed with the disease. There are therapies or processes to try to limit, but you're working at such a level that really kind of says we really don't know yet. We really, and maybe I'm not understanding that, totally, but it's just fascinating how much we don't know and how fast we are starting to know. Do you ever think about that? And I mean, I don't know if you've had any family members with any of these diseases, but it just seems like a really big separation.
Yeah, so it's been really interesting because I think on one hand, we are starting to know as a field a lot more about the genetics of these. So I would say even the last 2 to 3 decades, our understanding of the genetic causes of these diseases has exploded. And that's helped us sort of understand, give us clues into what proteins go wrong in these pathways. And so our understanding of, which proteins I think has gone up dramatically. I think the part that we're still trying to understand is what is the function of these proteins. So we know and one way I like to think about this is, if the protein is a person in the town, like what went wrong? Like how did they cause the fire in the town square? And there could be many ways. And, some of these proteins have many functions. So we also don't know like which function of theirs went wrong that ended up, causing the fire in the town square. But we are also understanding that, maybe there are new functions that no one's ever realized these proteins have. And so I think at the level of the cell biology and using live microscopy and sort of a lot of other tools available, the field is understanding a lot more about these functions. And I think the more we understand about the functions, we can actually figure out like what is the pathway that's gone wrong. And so one of the really interesting things from our research is that we're starting to figure out a lot of these new pathways. And based on these, actually, I think it's giving us really interesting clues that make sense, that bring us back to, I mentioned earlier that a lot of these diseases, they don't have a genetic cause. And what we're discovering is that as we learn more about the normal function of these proteins, it actually gives us a clue to what we think could be causing these different diseases in patients who don't have mutations.
Yeah. I love the description of the person in the town that, came from a good family and had everything, but yet set fire to a building. Like, why? And it does kind of put it in some kind of perspective of just how complicated or random this is probably happening. And do you think we'll ever find, why this is happening?
Yeah, I definitely think so. I think in a lot of these cases in Alzheimer's, Parkinson's, and ALS, I think the field has had so many clues. And I would say even in the last few years, our understanding has gone up a lot more, especially at the basic cell biology level. So I think we have a lot of these clues in our hands. And I think I'm hoping sort of, in the next few years, we'll sort of be able to nail what we think are like, this is the key pathway that causes it. And then to really test, if we target this pathway, is that actually able to help patients?
Yeah. Yeah. So, how do you see this whole field of neurodegeneration research evolving the next decade? You just gave us hope that yes, we'll probably understand it, but what do you see in 10 years, 15 years? Pick your time frame.
Yeah, I mean, I would love to see by that point, we've nailed the pathway. We know like these are the three steps that could be targetable and that we've actually figured out ways to target it. I've mentioned before that, these organelles are really dynamic. And so I think one exciting thing is that with the technology that we have now, I think we're actually able to figure out, what dynamics went wrong. I would say a lot of the past research in these fields have mainly just taken snapshots of the cell, which is of course really helpful. But if you want to know, like, how did the person make their way to the town square, if you could actually video that whole process and figure out, how did they set fire to it, you would actually know the whole process of what happened and where you could stop that process. So we're thinking, the more we learn about these different dynamics that change over time, the more we'll really be able to identify what went wrong. And actually, one reason we think dynamics are so important is because neurodegeneration is age related. And so it basically requires a lot of time, decades for any of these symptoms to have an onset. And so we think that, this gradual change of dynamics over time might not matter in 1 year, but over time they accumulate. And so I think the more we learn about, how these dynamics get messed up in a disease, I think we'll be able to have really interesting insights into the therapeutics.
So tell me about your lab. Like, how many people are in there? What is kind of a day in the life for Yvette's lab?
Yeah, so currently we have 4 PhD students and 3 research technologists. And so a day in the life of the lab, there's probably someone doing live microscopy. There's probably 2 or 3 people reading papers, analyzing their movies. And then, a couple of people looking at changes in protein levels, figuring out are proteins interacting with each other? Does that change in different disease conditions? Writing up our results. So lots of things going on at different times.
Yeah. Do you use, I should have asked this question earlier. So are you using human tissue and cells to look? Or do you have mouse models that you're looking, or both? What is kind of the source?
Yeah, we've tried all sorts of things. So, sort of our bread and butter to really figure out some of these like new cell biology pathways that no one's looked at before are using these human cell lines, HeLa cells, and so those grow really rapidly and they're really great for imaging. And so the interesting thing is actually, it helps that they are a human cell line because we found, there's obviously lots of differences between a human and a mouse protein for many of these proteins. But the other interesting thing we found is actually some of the things that you would see in more of like an adult cell, we can still see that recapitulated in a HeLa cell. And that's also been really helpful. But what we're sort of doing is trying to figure out these new pathways in a HeLa cell. Those experiments are pretty fast. And then once we think we've nailed the pathway, we've gone on to start validating those in human neurons. And so we have human neurons in culture that are iPSC derived. And we're looking at those with patient mutations versus not. And we've also had collaborations with other labs where we've looked at mouse models of different neurological disorders, where we also look at different organelle dynamics in those neurons.
You mentioned how you can see these pathways. And then I think you mentioned with like Alzheimer's, it's age related, but it's also kind of the journey of life related. How would we study, or are we studying kind of those pathways through maybe somebody that's younger, 30, 25, 30, to that age where we kind of tend to know when Alzheimer's starts to really be visible in someone?
Yeah. So I think this is sort of maybe I would say a challenge of the field. How do you accurately mimic or model a disease that, can have an onset of between 40 or 70 years in an experiment that are not going to do for 40 years, hopefully in less than 2 months if possible. so, um, so one of the ways that we try to model this is sort of accelerating it using mutations that we already know cause the disease. So that sort of pushes the system a little bit more. But we've also found recently that there are a lot of other factors, very different things for each disease, whether it's mitochondrial function, the type of lipids, the abundance of lipids, all of these different things we think might be contributing to different aspects of each of these diseases. And so we can actually sort of artificially accelerate the disease by increasing those levels. And that is actually one of the clues we have that suggests, hey, we're maybe actually accurately modeling it if we can, push the system a certain way and actually see the phenotypes in a neuron that you would see in an actual patient, but, 70 years later.
Yeah. So you mentioned, that's a challenge. What other challenges? I mean, we have the normal. I would assume the normal challenges of, finding talent and making sure you have enough funding and maybe even time on the technology, because I'm sure these microscopes are expensive to use. But what other challenges do you have, especially as a younger leader, earlier career, if you would, leader in the lab in kind of today's research environment? And I'm not talking the political part. I'm just talking about the accelerated time and kind of demands on science and research.
Yeah. So I think science is always changing. And so I think maybe one interesting thing, which is actually kind of a helpful thing, is that, the rate of science is really fast, which is great. It means, we're making more discoveries and like bigger discoveries. And so at a faster pace. And there's also a lot more people who are doing science than ever before. And so I think one aspect is sort of like keeping up with what has been found. And if you look at sort of the number of publications published, it's like exponential in the last decades. So there's just a lot more information to know, and figuring out, which of this information that's coming out is important or helpful to what we're studying. So I think that would be one angle, sort of keeping up with the literature and figuring out which of those is important and relevant to our disease we're interested in. And I think maybe related to that is sort of, the people studying on these different diseases are sort of all over, and how do you sort of keep up with the latest thing in each disease field? And so I think on one hand, it's actually really great that there is, the advancements are going this fast, but I think just knowing how to incorporate that into our research is something that we're always thinking through.
Yeah. Yeah. How do you do that? I mean, I've delved into PubMed and Google Scholars and there's so much. And then the question that even if I could kind of grasp some of the reports, was there somebody that found something that countered that paper, or what is that evolution of kind of the science? Has it been proven differently just a year or 2 year later? How do you keep up, and then how are you kind of mentoring your PhD students and to keep up?
Yeah. So one thing I actually do is at the beginning of the week, I always set aside time to look through all the relevant papers that are in each of these diseases and the cell biology pathways that we're looking at. So obviously, as there are more and more papers coming out, this takes a little bit more time than it used to. But that has always been helpful, and this is helpful because it helps us know what's the latest thing, but it also keeps us sort of on our toes to think like, oh, is there a new hypothesis that, or a new clue that we can add to our story that will give us insight into like the next step of this pathway. And so something that I will do is, if I see new papers that come out that are relevant to one of my grad students' projects, I'll immediately send it to them. But we also have like group channels in the lab for them to sort of let each other know, oh, did you see this other paper come out? And so it's been really helpful for not just me to share this, but also I think it's even more exciting when it's the other lab members who are sharing with each other things that are relevant for each other's projects.
Yeah, I think that's great because I think, AI or these tools can only do so much in aggregating. It really takes the people that have the deeper understanding that human communication seems to be really important.
Yeah, that's right.
Yeah. So you received the NIH Director's New Innovator Award. Tell me about that and what was that for? And how big was that? I mean, not big, like financial big, but how important was that to you?
Yeah, I was very thankful for that. I got that right when I was starting the lab. And so that basically helped us to be able to have more people join the lab. And also, it was definitely helpful for getting our research project started. And that was sort of an angle that we were interested in. How can we leverage this super resolution microscope to look at what's going on inside organelles? So far, I've mainly talked about these interorganelle contact sites when two different organelles come together. But yeah, with this, we were hoping to sort of see what goes on inside an organelle. And we have some really interesting findings based off this. So it's been exciting.
Yeah. And have you published on that yet? Or
Yeah, so we're trying to write up some of the things that we found from this. So hopefully, in the next few months,
that's
we'll
great.
have some things start coming out.
Yeah, that sounds it's probably a pretty prestigious award that I mean not a whole bunch of people get that. Is that true?
Yes, that's true. I think they have a certain cohort that they give this to every year across all of biology. So I was very thankful for it.
Well, that's amazing. That's wonderful. And I know we've given you the Karen Toffler Charitable Trust has given you a grant, and I just want to ask, how important is that? Because that is kind of unconnected to any. It's really what you want to do. Like you don't have to come with any proof of anything. You can do something that's high risk that may not pan out on from a hypothesis. Can you talk about that?
Yeah, this was really exciting for us, and so we're really appreciative of this. We have a lot of projects that have started in the lab that were things that we did not expect at all. So when I first started the lab, I was excited about contact sites, I was excited about mitochondria and lysosomes, and I knew I wanted to figure out more of these pathways in neurodegenerative diseases because I felt like there were still a lot of questions that were not really answered at the mechanistic level. And I would say that's how we started. But from there, the most exciting studies we've had are literally when I have someone in the lab come and tell me, oh, I think I saw something interesting on the microscope that I'm not sure if that was what we were looking for. And those have been really cool because all of those have spawned into, these projects we didn't even plan for. And so we're really thankful for this Karen Toffler Charitable Trust Award because it's really helping us look more into these other questions that we think actually also have some really important clues back to both cell biology and into neurodegeneration that, when we started out, we didn't even think this was a project because we didn't even know it existed. And so that's actually one of the wonders of microscopy. You can be looking for something else, but and then you suddenly see this really interesting thing that literally nobody in the world has seen before. And so it's all of these processes have been really fun to try and figure out what are the mechanisms, what's driving this, how does this go wrong in disease, how many clues can we get from what other people have published, from what we know about disease mutations, and sort of at the end of the day, can we help patients if we figure out the pathway that's actually really important in a disease?
Yeah. Yeah, that's great because, science, it can be very serendipitous in a lot of cases. How do you write a grant on something that you don't even know exists yet? And so I think that's wonderful. When I don't know if you have friends, family, acquaintances that aren't really close to research, in the kind of the research ecosystem. But what are some of the kind of interesting questions that they ask you when they find out what you do? I mean, I would imagine it's kind of interesting to hear what their questions are and just to see maybe, how far some people are from research.
So sometimes the questions I get are, like what is a protein, or what does a mitochondria do? But I also get questions like, exactly like ones you asked, like, how do we actually model a disease in a lab? Do we think we're modeling the right thing? And also, what sorts of things can you look at with a microscope? So for me, it's always just been fun to kind of explain, what is it like doing biology research, and I love explaining it to other people. And so for me overall, it's just fun to be able to share our research and also to try and explain it to people who aren't normally doing research.
Yeah, it's really fascinating, and I bet it is pretty fun. So, we talked a little bit about the future around the diseases and what we might learn. But, as you just kind of peer into the future 10, 20 years from now, you'll be this amazing professor that has a lot of runway behind you of success. What kind of breakthroughs or what kind of significant things do you hope we've achieved?
Yeah, so I think the field of neuroscience as a whole has a lot of really interesting things going for it right now. I've talked a lot about like specifically neurodegenerative diseases, which I think has, a lot of exciting things. And I'm hoping sort of, in the next decade, we'll be able to crack at the mechanism level, figure out sort of what's going on there and how to target it. But I think overall, when we think about neuroscience, I think with things like brain organoids, we're able to study a lot of these connectivity and questions that were harder to study in a human brain. So I think that's one angle. I think there's a lot of interesting imaging at the whole brain level that's starting to be done at an even higher resolution level. And so that sort of combined with these different omics approaches out there, I think we're understanding a lot more about how the brain works and what the different regions are, and how do you mechanistically target different brain areas. So I think advances in any of those areas would be really cool. I think it would also just be really interesting if we can even figure this out sort of at the dynamics level. So, in sort of my dream experiment, one day we could tag every single protein simultaneously and, watch them all moving inside the brain in every single neuron and know, in this condition, if your mitochondria aren't working, where do all those proteins go to? Right now, we're sort of, tagging 3 or 4 at the same time. But if we could tag every single one and track those dynamics, I think we would learn so much more than we already do.
Yeah, that would be amazing. Maybe the advanced technology, machine learning or AI or some of the supercomputers that we'll be building in the next 10 years will allow us to do that.
Yeah, that's right. And there's already a lot of, I think, hints from exciting technology, even in the last 5 years, that suggests, there may be steps we're taking towards that.
Yeah, that's really cool. Well, Yvette, you are so good at this. You are so good at communicating what you do on something that's very hard to even understand. I can't see these little things moving around, and none of us can. So, thank you so much for the visuals. As we kind of wrap up here, how can people that are curious about what you do, or want to collaborate, or want to fund you, how would they get a hold of you?
Yeah, so you can check us out on our lab website, Yvette Wong Lab. If you Google that, you'll see our lab website. You can also directly email me, yvette.wong@northwestern.edu.
It's been a real pleasure. This is not the first conversation we've had, but you just make it so clear, and I really appreciate your time.
Thank you so much. I really enjoyed this.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.